Good morning, everyone, and thank you to Mr. Somani, Mr. Sood, and all my friends who are here. And thanks for inviting me here and giving me an opportunity to speak about our work in my hometown. I'm going to talk about two strands in our work. One strand of work is in neuroscience, and most of this work happens in the laboratory. And the other strand is outside the laboratory, which could be perceived as an applied version of our work, but mostly it deals with greater issues and much bigger challenges. And as such, I have called this talk Autism Research in and Outside the Laboratory, but they are targeting two different types of challenges. The first type of challenge, or the first type of barrier that we are up against, is the barriers within the brain, and this line of work is the research in the laboratory. In discussing barriers within the brain, the particular problems that our team has been focusing on are those of social cognition, of social understanding, and social communication. Atypical social behavior is well documented in autism. There have been several theories over the last several decades on this. Prominent among those are those that have suggested there is a deficit in theory of mind which allows people to understand other people's intentions and mental states. This theory that came up in the 1980s then gave way to a larger more broad-based theory of a deficit or atypicalities in empathy. Empathy being the ability to understand and respond appropriately to other people's emotions and mental states. And of course, while the psychologists were coming up with these theories of what is the core social feature of autism, the neuroscientists, armed with their new technologies, particularly brain imaging, were trying to look for neural signatures, brain-based signatures of these social behavioral features. One needs to remember at this point that finding a brain equivalent of a social behavioral feature doesn't make it any more or any less real. There has been, and there continues to be, some understanding or some misunderstanding in the public that if we can find a brain signature, that suddenly makes autism more medical, more real, more curable of sorts. That is not the case. If there is an observable behavior, there is most likely going to be a brain signature. So in case of autism, the brain signatures have tended to be focused along a set of regions that came to be known as the social brain, and which includes various regions that are marked here along the temporal lobe, the superior temporal sulcus, amygdala, and the orbitofrontal cortex. But all of this really comes from the psychological model that there is an empathy deficit in autism. But is that true? So the first point of my talk is really asking whether the empathy deficit is an epiphenomenon, by which I mean, is the empathy deficit a product of a more fundamental feature that characterizes the autistic brain? And this fundamental feature is one that is driven by reduced responsivity to social rewards. So what we think, or what we hypothesize as an empathy problem, is actually a reward-based problem. And this is the line of logic that goes toward explaining it. So if somebody does not prefer to look at faces and social stimuli, then if you're thinking of it from very early on in childhood, if the child is not attending to social stimuli because of a lack of interest, they are unlikely to learn from social stimuli. If they are unlikely to learn from social stimuli, then they will not figure out various rules of expression and not understand the various nuances of facial expressions, for example. And then by the time we are assessing things in later childhood or even adulthood, we might mistakenly assume that the problem is one of empathy, but where the seed of the problem is actually one that's based on a typical reward responsivity, that initially the reward responsivity was not there, and hence we see all these deficits in understanding and responding to other people's emotions. In this context, we have shown several studies, we have done several studies showing the link between empathy and reward processing. So in this particular study that was published a year and a half ago now, we showed people pictures of social rewards like facial expressions of happiness and people jumping up with joy, and food rewards, non-social rewards. And we were measuring how long people looked at one or the other kind of reward when they're presented simultaneously on the screen. We are recording their eye movements using an eye tracker. And what we find is that people who are high in empathy, they tend to look much longer at the social rewards compared to the non-social rewards, suggesting that what we measure as trait empathy is very much related to how much you prefer looking at social stimuli. So this preference is quite distinct from, or possibly precedent to, understanding and responding to social stimuli. Now, this study was done in adults. Again, a year and a half ago, we did another study where we made a tablet-based gamified task where people had to press, these are kids, 15-month to 40-month-old kids, they have to press one of these two buttons. These are jumping buttons, and if they press one of these buttons, it reveals a face of a smiling child. If they press the other button, it reveals a train. And what we found is that toddlers with a diagnosis of an autism spectrum disorder selectively pressed the button that led to the social stimulus less than the number of times they pressed the button that led to the train. Interestingly, though, the typical toddlers, the toddlers who did not have a diagnosis, they did not show a greater preference for the train over the face or the face over the train. So those are the figures for the typicals, whereas for the autistic toddlers, we have a selectively reduced preference for social stimuli. One more study from adults where we used a technique called continuous flash suppression. Now, all of the studies, the two studies that I talked about before are where the social reward and the non-social rewards are presented well above the threshold of awareness. So they can see it for several seconds, and so there is a lot of time for the brain to process it and respond to it. But what if you present the stimuli, the social and the non-social rewards, below the threshold of awareness for very fleeting seconds, or by manipulating using this technique called continuous flash suppression that I'm happy to talk about after my lecture, where these stimuli are presented subliminally, and even there we find that adults with autism do not show a preference for social stimuli that we see in typical adults. That typical adults are usually much faster to detect social stimuli, social reward stimuli that are presented below the threshold of awareness. But we do not see this preference for social stimuli in adults with autism. Again, showing that there is a very close connection between reward processing and empathy-related abilities and autistic traits. These are just a few studies in autism, and we also have a whole body of literature from animal models and other human studies that show a link between reward and empathy. Animal models, particularly studies in rodents, in rats and mice, have shown that if we can take out the brain regions that are processing rewards, either surgically or pharmacologically by putting in an agent, if we block those reward processing regions, then the animals not only, I mean, they don't have other major issues, but they have huge deficits in how much social behavior they will exhibit towards their conspecific. So how the mouse mothers will behave towards their pups, where they'll stop licking, grooming, and nursing their own pups if their reward centers are either surgically removed or blocked pharmacologically, suggesting that there is, again, a close connection between reward processing systems and what we think of as social behavior and empathy-related behavior and animal equivalents. We have also shown several human studies with showing a link between these processes. But all of these studies that I've mentioned so far are dealing with empathy as a big construct. Empathy usually measured with trait questionnaires, and but it's not one thing. Empathy is not one monolithic construct. So we need to delve a little bit deeper into what empathy actually consists of. And here we see that if there is one, so the person on the left is the person expressing an emotion, and the person on the right in this model is the person observing the emotion. And all of these processes are component processes of empathy. So the first part is perception. So just this person on the right needs to see that the person on the left is expressing an emotion. That is then followed by some kind of motor mimicry or embodiment into this person's own motor system. Then there is a self-other distinction. Then there is an inferential or cognitive process suggesting that that tells this person that, okay, why is this person feeling like that, and all of that. But one of the crucial processes within this set of empathic processes that we talk about is motor mimicry. It's a very early process. It does not require the cognitive resources that one would need as an adult to understand another person's emotion. And it is seen also in infants. And if I can show proof of that, there might be this video. Let's see if this works. So I hope that would assure you that they are not engaging in any theory of mind or anything that requires any cognitive abstraction. This is fairly basic, happening very early on. And it is this specific process within empathy that I'm going to talk for the next few minutes on, that we have now focused our investigations to. So just to kind of a quick recap, the first few studies that I've talked about have suggested an overlap between the reward and the empathic processes and how they may be relevant for autism. And now I'm talking about a specific process within empathy, motor mimicry, and how that interacts with reward. So I'm narrowing down. Now, we know from various psychological studies that mimicry and reward have a strong link. People who you like more, generally you tend to mimic them more, and vice versa. If somebody mimics you more, then you tend to like them more than somebody who doesn't mimic you. And more importantly, if you block mimicry, if you block the facial mimicry, then you become worse at detection of facial expressions of emotion from other people, because that blocks that process of motor mapping that is very important for understanding another person's emotion. What do we know about mimicry in autism? We know that it is reduced in magnitude. We know that it is slower. However, crucially, there is no obvious deficit in deliberate facial mimicry. So if you ask people with autism to mimic another person's facial expression, most of the time, most of the people do just fine. However, in spontaneous situations, that often is not the case. To measure spontaneous facial mimicry, what we use is called facial electromyography, where we put electrodes, as you see in this picture, on the relevant muscles of the face. So here you just see one muscle, the zygomaticus major, that is involved in smiling. We also record from various other muscles, particularly the corrugator, which is involved in frowning. So what we asked was whether reward modulates the extent of mimicry. Does it matter that I am looking at a rewarding person versus a non-rewarding person, and whether in how much I will mimic them? So if I'm looking at my best friend versus if I'm looking at a complete stranger, am I more likely to copy mimic my best friend? From folk psychological theories and from common sense, one would say, yes, that's what's supposed to happen. But it's surprisingly, when we were starting on this line of work, there was no data that showed this in any scientific way. So what we ended up doing was a reward learning experiment where participants played cards against they were playing cards, and they had to guess whether the card that they cannot see, whether it is higher or lower in value. And if they guessed right, then they win 25p. If they guess wrong, they lose 20p. And they were told that every time they're playing this card game, they'll see some faces coming up on the sides. Please pay attention to those faces, because there will be a memory game with the faces later on. And what ends up happening is we actually manipulate the presentation of faces such that, for example, every time you see this face, you end up winning about 80% of the time, and with other faces, you end up losing 80% of the time, so on and so forth. So you end up 4 faces that are conditioned with high and low reward values. So this, for this case, this gentleman in a yellow t-shirt, you will win 90% of the time against him and lose 90% of the time against the lady in red. And after these faces have been conditioned with high and low reward, I mean, the rationale for doing it is that we need to control the extent of reward associated with each face, which we cannot really do if we say, well, I'm showing you your best friend's picture, because one person's best friend is not equivalent to another person's best friend, and how much they like and how much they are rewarded by. So we need to make it quite exact in terms of the strength of reward. Now, after these faces have been conditioned with reward, then people watch the same faces, these 4 faces, making expressions at them. So either they're smiling or they're frowning, and what we are recording is the extent of spontaneous facial mimicry. So we are going to record the spontaneous facial mimicry in response to this high reward face, and also the spontaneous facial mimicry in response to the low reward face. And what we find is that there is significantly higher. So on the left panel here, my pointer doesn't seem to work very well. On the left panel here, you have the top line trace that's the extent of facial mimicry for the high reward face, and the bottom trace is for the extent of facial mimicry for the low reward trace, for the low reward face. We saw that, we see that in all individuals. Interestingly, this difference that you see in high versus low reward faces is seen only in neurotypical adults, and there is no difference in the spontaneous facial mimicry between high and low reward faces for people who are on the autistic spectrum, who have a clinical diagnosis. So this reward-dependent modulation of facial mimicry does not seem to be happening. Now, we then repeated the same experiment in the MRI scanner, and we find that again, in people with autism, the reward value of the face does not modulate the brain response to high versus low reward faces in people with autism. So here what we measure is the two areas that are marked out there. One of the blue circles mark out an area which is involved in processing rewards. This is the ventral striatum. The yellow circles mark the areas involved in mimicking, particularly the inferior frontal gyrus. What we are measuring is the strength of coupling between these areas. What we find is that in typical individuals, this strength of coupling varies depending on if you're looking at a high reward phase versus a low reward phase. But in adults with autism and those with high autistic traits, you do not see this reward-dependent modulation. We did the same experiment again now with EEG and measuring something called mu suppression, which is a measure of something called mirror neuron activity, but keep the mirror neuron aspect of it to one side. But effectively, all three of these studies are showing the same thing, that the fundamental issue about mimicry-related neural responses or mimicry processes in autism is not about whether there is mimicry or not in autism. It's whether the mimicry-related processes are modulated by reward or not. So the model to understand this is what I call the thermostat of empathy. So autism does not have a problem in empathy as such. Autism has a problem in modulating the empathy depending on the reward value of the context. And reward signal, so just like temperature will, the ambient temperature can change the level of the thermostat, the thermostat has to constantly calibrate, the reward value of the context, who I'm talking to, has to calibrate the level of empathy we will express. So that's one aspect of, that's the interim conclusion from this set of studies. In the next study, we asked this same question in the opposite direction, where we asked whether if somebody is mimicking me more, then do they become more rewarding or not? Again, in a very controlled experiment, in a laboratory experiment. I will not have time to go through the details of the experiment in light of the time. I'm happy to discuss this later. But long story short, again, we did another fMRI study and an eye tracking study where we showed that the reward-related brain activity in individuals with autism was lower in response to faces that were mimicking them compared to the faces that were not mimicking them. So to step back for a moment, individuals who, in the neurotypical individuals, show a greater reward-related response to mimicking faces compared to non-mimicking faces. Individuals with a diagnosis of autism do not discriminate between the mimicking versus non-mimicking faces as far as the reward value of the face is concerned. So the summary from this set of studies is that mimicry and reward work together like to function as social glue. If something is more rewarding, it will increase mimicry, and if somebody is mimicking more, then they are likely to increase reward. And this is how rapport, interpersonal rapport, is built up. So if you think, and what we see in autism is there is a weakening of links in both these directions. Both the reward to mimicry modulation as well as mimicry to reward are weakened links. And if you think of it from a developmental perspective, a lot of early parent-child interaction involves reciprocal imitation. And reciprocal imitation will falter if being imitated is not rewarding to the child. And if being imitated is not rewarding to the child, if the imitator is not gaining in reward value, then that will not stimulate greater mimicry. So this is just kind of unpacking the mechanism of potential areas through which the rapport, the interpersonal rapport between social interactants, and where one of them has a diagnosis of autism, can be looked at. So this summarizes the kind of the neuroscience aspect of it. There are lots of implications for it. I mean, if you think of the therapy, a lot of autism interventions rely on reward learning, including ABA. relies on reward learning and expects that the learned reward will generalize to other kinds of social behavior. But if we see that there is a definite weakening of the reward to mimicry link, then that might explain why these interventions do not work all the time. It might have to do with these connectivity between reward and mimicry related brain areas. Now, in the final part of my talk, I'll talk very briefly about barriers outside the brain. And here I would talk about something that's been called the detection barrier, which is the barrier of finding and getting an appropriate diagnosis at the right stage so that interventions can begin. Gauri, who is in the audience, and Vikram Patel have pioneered this work in various parts of the world, and we have done this work here in Kolkata. So I'm going to very briefly talk about some of that work. The detection barrier is very important in getting early intervention, and one of the key existing barrier there is the lack of availability of good screening diagnostic tools in local languages and lack of clinical experts in mental health. Now, about 10 years ago, Shanil Mukherjee and I, we were, and Dr. Ram, we were sitting around, and we wanted to do an epidemiological study of autism in Kolkata, and I thought that we will just copy the British way of doing it, that we will find the outpatient records from the various hospitals around the city, and that will tell us the prevalence of autism in India, and or at least in Kolkata. And we realized that most hospitals, and these were mostly government hospitals, did not have a register of outpatient records of how many came in and how many left with a diagnosis of autism. So we immediately had to change tack and had to move to a population-based design where we would go into schools and screen children. And for that, we needed to translate and validate the screening and diagnostic tools for autism. So at the state of play in 2014, up to 2014, was that these were the different prevalence studies happening across the world, and India was kind of marked by the absence of a population-level prevalence study, and whereas most other parts of the world had loads of them already. So I don't need to explain how many people speak these languages in our part of the world. So our first studies were on translating and validating some of the gold standard tools used across the world on autism diagnosis and screening. These included the SCDC, AQ, SCQ, and ADOS, as well as behavioral and cognitive measures like reading the mind task, emotion recognition, and embedded figures task. This is all data collected from Kolkata, again showing what you see here is the blue and the red markers are all the two groups of children with and without autism. These have been validated in two sets of papers in 2014 and 2016, led by my PhD student Aloka Rudra. And then all of these studies kind of culminated in a prevalence study of autism in 11,849 children from 30 schools of Kolkata. This is this is 28 schools plus non-government, the Anganwadis as well. And these are schools across the socioeconomic spectrum. What we found was a prevalence rate of only around 0.26% in this sample, and none of these children who we diagnosed had originally a diagnosis of autism. So what this suggests is two things. One is that we are possibly just seeing the tip of the iceberg because we are sampling only from schools, and we are not sampling from children who are not going to school, so we are possibly missing a much larger segment. And two, a lot of children so 0.26% appears low, but once you multiply it with several million children that are around in mainstream schools, there are a lot of children who would qualify for an autism spectrum diagnosis in mainstream schools, even if they don't have one right now. So there are some pictures. This work, I have only half a minute left now, and this work has now led to our current work where we are developing an app which has multiple aspects of autistic phenotype that we can measure. This is a collaboration between several universities and NGOs across the world, and especially in India. And here is the app website for those who are interested. So we have one of the key things is that we have measures that have come from the lab. So on the first part of my talk, when I talked about the reward sensitivity, some of those measures, we have now translated them from the lab into the tablet, and we have used tablet-based eye tracking. This is a new technology where we are using the front camera of the tablet to do eye tracking rather than an eye tracker that we use in a typical lab-based study, to look whether people are looking at a social reward more or a non-social reward more. You remember my smiling face versus chocolate slide from the beginning, and that's helped us look at whether people are looking at stimuli that they prefer or they don't. And the crucial thing is that this app does not require a specialist to administer it. So it would be community health workers with minimal training who would go into the household of children, and these are children, usually 3 to 6 year olds, that would be administered on, and we are collecting the data. Again, Gauri's team from Delhi has collected data from about 120 children with autism, with intellectual disability, and typically developing children, and we are seeing some very promising results in group differences that that acts as proof of principle for this app working. And so here is a quick example of one social task and one Visuomotor task from the app. So this is based on the gamified app that I talked about before, where the children have to press one of two buttons to see a social or a non-social stimulus, and we measure how many times you press the social button or the non-social button. That's the button task, and this is a task where children have to trace a bee that's flying across the screen, and we are looking for motor coordination issues. I'm sorry, the trajectory is not very clear here. And indeed, that brings me to my final slide. These are all the people I am immensely thankful to for the work that I've reported here, plus the people who have taken part in our work, and my funders. Thank you.